All right, so we're going to get started today. Thank you everybody for coming. I just want to first start off introduce myself to my classmates. My name is Tom Manny. I work for Monsanto in Stonington, Illinois. Halfway about between from Springfield and Decatur. So with further ado, we're going to get started. My presentation is covering a pretty interesting, relatively new piece of technology called CRISPR-Cas9, and which a lot of people believe is going to be the next step in biotechnology. So we'll go ahead and get started. So before I get started here, before I start talking about the nitty-gritty, to appreciate where we're going in biotech, it's important to take a step back, look at where we've been as a society. So I'm going to start off with a brief history of plant breeding, some of the key points going on through the time here. So we're going to start off, it all started about 10,000 years ago when sedentary agriculture began, which is when humans began to live in communities and start to grow and produce their food from, in their community, rather than being a nomad type society where they traveled across the land looking for, hunting game, gathering different fruits and vegetables, much harder type of life. And so this is really when, human society has started. So, very important here. And even though they didn't know it at the time, with sedentary agriculture, that's when selective breeding first began. And a great example of this, as everyone has seen, is teosinte. Teosinte is the early parent of modern day field corn. We've all probably heard about this. Is through the years, ancient civilizations in the Latin America, North-South America region began growing teosinte as a grain crop. And through the time, they would start selectively breeding varieties that they wanted. Important traits they're looking for, grain quality, so they don't want something that's going to rot and get moldy, because that's bad. It's a food crop. We want it to be, good, hearty, and nutritious. Next is, seed dispersal. When you're looking at a native crop, a plant's natural, it's naturally looking to spread their seed. Look at dandelion. Those things blow all over the county. So they want a seed dispersal. So it's important to reduce that down in a crop so the grain is staying with the plant so it's easy to harvest. And then, finally is yield. That's still a major component today. They want to grow something that's going to get them as much food possible with the least amount of inputs. So we'll go ahead and move on here. Next came about 8,000 years later or so with Gregor Mendel, known as the father of genetics. We've all probably filled out a Punnett square back in high school biology, and if you're anything like me at the time, I thought there's no way I'm ever going to use this. What am I going to do with this later in life? And sure enough, here I am, 10 years later, giving a presentation about them. So it's kind of funny to see where you've come and where you are now. So anyway, Gregor Mendel, we all know his his research in peas. So he would start crossing pea plants and looking at different genetic traits and whatnot. He published his works, Laws of Inheritance, in 1865, and an interesting fact about that is that his research wasn't significantly, the significance of it wasn't recognized for about 3 decades later, until about the turn of the century, when people decided to really start using his work and applying it to modern agriculture. So on the left is a picture of a pea plant here. On the right is the Punnett square that I explained, and it's important here, so oops. There we go. The basis of a Punnett square is still very relative today when it comes to population genetics. So just to kind of break it down a little bit, we cross 2 pea plants here. Both have a purple flower, and both are heterozygous. When you cross the 2 together, you're going to get a 50% chance that they're going to be heterozygous right here and here, and a 50% chance that they're going to be homozygous right there and there. So knowing that, if you're making a cross in modern breeding something of your own, it's important to know that if you're looking for something like a purple flower, which is a dominant trait, you have a 75% chance of a cross being purple flowered and a 25% chance of it being white flowered. So it's very important in applying this to, okay, if you want to get a certain amount of plants with a trait in it, this applies very much so, of knowing how many crosses you should make and what your percentages are that trait is going to take onto the plant's progeny. So with that, we'll move on to the next part, which is the Green Revolution. The Green Revolution was a period in agriculture when there was a significant amount of advancement, going from about the 1930s till the late 1960s, primarily led by Norman Borlaug on the left here. And because of this, they started, a lot of new technologies have been implemented. Technologies such as irrigation, pesticides, synthetic nitrogen fertilizers. Those are the big ones, but also another part of it that we don't really, a lot of people might not recognize of it, is the the massive advancement in plant breeding knowledge. So we've they began to learn about hybrid crops, learning that the offspring of two inbreds can definitely show what's called as heterosis, which is where the offspring is much more vigorous than the parent crops. And that is very important. So, oops, one second here, I apologize. So with that, with the knowledge of heterosis, they began to produce very high yielding cultivars, which significantly increased the population through that period. Somewhere in the marks between 3 and 4 million people grew between 1920 and 1960, as you'll see here on the next slide. So as you can see, 1920 here, populations globally remained relatively linear, and marking the beginning of the Green Revolution, you can see that the population began to grow exponentially. Right now, we're sitting at about 7.5 billion, and this slide is projected through, well, shoot, I don't even know how to say that, it's so far out, 2100, I don't know if that's what we're going to call it then, but what happened here? There we go. I must have hit a button. Apologize again. Anyway, 2100, it's projected to be over 10 billion. So in order to do that, we're really going to have to gear up our ability to produce food. We all know that we have to feed more people on less inputs. And new technologies such as biotech is going to drastically increase our ability to do so. So moving on. So now I'm going to move on to some points in modern plant breeding, where we are today, and what sort of technologies we're using. So the first one is marker-assisted selection. Marker-assisted selection is when you can screen a plant for certain desired traits that you possess markers for. So if you want to look and see if a plant possesses the Roundup Ready 1 gene, you can send it to a lab, it runs it through the markers, and you can see, all right, this plant does in fact possess that trait. So this is easily done with transgenic traits because we know what those markers are. We know the sequence that we put into those plants and what to look for. With traits such as natural resistance to different diseases, it's a little more difficult. Markers have to be developed and found and used that way before you can begin to use that, those markers. So, oh god darn, I keep hitting the wrong button. So next are DHs, or double haploids. Double haploids is a genotype formed when haploid cells undergo chromosome doubling. And so this is important because in order to get a genetically pure inbred, it takes about 6 generations. With double haploids, you can get that in 1 generation. So that drastically reduces the amount of time needed to get that perfect inbred for what you're looking for. It saves a large amount of time. And we all know if we work in private industry, time is money. So that is very important. And then finally is genetic modification. It's taking a trait that isn't naturally present in a plant, inserting it in, and then you're allowed to use, any sort of, gene, trait, whatever you want, something that could improve that crop's ability to produce. So through genetic modification, a lot of people, including myself, didn't really know. I knew that there's Roundup Ready 1 genes and Drought Guard and all these other different cool traits and crops, but I didn't really know how they got into it. So I started looking into it and to figure out what it is. So how it's done is, so scientists will identify the donor gene. So for this example, we're going to say Roundup Ready 1. So you identify the gene that you want to insert into a soybean plant. Then you use a couple different methods. You can use a gene gun to quote-unquote bombard the plant, the target species, soybeans, or you can use an agrobacterium, which kind of naturally infects the plant, creates sort of like a cyst, and the gene is uptaken that way. Two different ways to do it. Both can ultimately result in a plant possessing the gene that you're looking for. And then what you have to do after you get that, after you initially, I guess you could say, bombard the plant with the target gene, you have to screen those plants out, see which ones have actually taken the desired trait, see which ones has it in the best place, and there can be multiple outcomes. One plant could have could possess the trait on 10 different chromosomes. Well, you don't want that because that's going to affect too many different things in the plant. But the also scenario is if you bombard 10 different plants, then you could get 10 different locations on 10 different chromosomes of where this trait would be. And then from there, you want to screen it out and see which location of the trait affects yield or any other affects the plant phenotypically in any way. So that's what you're looking for when you're screening. So the problem with that is it's very expensive. There's lots of money spent in trying to get these genes to uptake, trying to screen them out and see what the best location is for this trait. And there's really no control of where it goes. You're going to get whatever cards you're dealt. So, as I said, this method it works. It's produced hundreds of different, very successful traits, but it's not necessarily the most efficient way to do this. And with that, I'm going to dive into CRISPR-Cas9. This new piece of technology that was developed in 2015. A little controversy kind of going on with it. I'm just going to say all parties' names. So it was developed by UC Berkeley in conjunction with the University of Vienna, but also the Broad Institute in Boston. Currently, the U.S. awarded the patent to the Broad Institute, but that's currently going up against an appeal. UC Berkeley took them, so it's going to be a while before we actually know who owns the patent to this technology. But in the meantime, those two parties are still licensing out this technology. We're going to deal with that later. Companies such as Monsanto have already licensed this technology and are allowed to use it. So what's nice about CRISPR-Cas9 is it's easy, it's customizable, it's precise, and it's cheap. It's much cheaper than the old way that we were doing it, and it can be applied to any sort of biological organism. So plants, as I'm talking about here, animals, they use it in labs to kind of even the playing board on different mice for testing. And the scary part is even humans. I'm not going to dive into it too much. It's a touchy subject, but if you have done any research on CRISPR-Cas, you may have heard the term designer babies. Currently, there's a big, there's some discussion going on about that. So now I'm going to explain how CRISPR-Cas9 works. So CRISPR actually has a pretty easy acronym. So what it stands for is clustered regularly interspaced short palindromic repeats. So I expect everyone here to memorize that before we leave today. That does possess a meaning. I'm not going to dive into it too deep because it's a little cumbersome. But what CRISPR is they're segments of prokaryotic DNA containing short repetitive base sequences. And so how this system was found is that bacteria possess this CRISPR system, which is when they're infected with a virus, they actually absorb part of the virus's DNA and place it into their genome, thus giving that bacteria a resistance to that virus. Kind of similar to like a flu shot. You get injected with the flu shot, and for the year, you could be safe from the flu, from a certain strands of the flu that could affect you. It's very similar to that. So, I keep doing that today. So, as I said, CRISPR are stretches of genes from the bacterial genome, and the Cas genes are closely related to the CRISPR genes. And what Cas does is they encode proteins which carry the DNA sequences. So, in the two DNA sequences, there's two strands. CRRNA, CRISPR RNA, is the actual map. That is what Cas9 proteins carry throughout, going through a plant's genome, looking for the CRISPR RNA sequence. And when it finds that sequence, it cuts the it can either make a single or double-stranded cuts. In this example, we'll just say it's a double-stranded. It'll cut the DNA there, and then from there, it's going to do one of two things. The DNA is going to leave a gap, or the DNA is going to repair itself. This is advantageous because companies can produce their DNA, their genes that they want inserted into a plant, and they can be carried along through this Cas9 protein. And then when the Cas9 makes the cut, the gene that the company has targeted will be used as like a model for the DNA to repair itself. So this is kind of how the system works. A little cumbersome. I know there's a lot of new things going on here. Best example I like to think of this as, so you're at a ball game, you're going to downtown St. Louis or something, and you park in a parking garage. And what you want to do is, your goal is you're looking for a spot. Cas9 is your car, and you're going to drive serpentine through a parking garage looking for that open spot. So that's my best example of how Cas9 works. Goes through, you find your spot, you pull in, you park, mission complete. So that's an easy way to kind of analyze and see, okay, this is what this is doing here. So, where did my mouse go? All right. So anyway, next slide. So now I'm going to leave the future of CRISPR-Cas9. So what this allows for, it allows for a quicker way to get genes into a target species. It's going to allow companies to implement new traits if there's a devastation in a certain part of the world, where with a disease that is actually wiping out a country's food plot or food source. CRISPR is going to allow you to companies to insert a certain resistant gene into a genome that's not going to affect things. It's not going to take as much time, and ultimately it's going to be able to feed people a lot faster and kind of allow us to launch us into this next millennium and keep us around for a lot longer, allow us to keep us fed on less acres. Another thing for corporate companies is it saves a lot of money. You're going to be able to use this. You're going to be able to get species, new traits to societies a lot faster. New things, new defense mechanisms. The door's wide open for what we can do here. It's pretty exciting, pretty interesting stuff. Currently, there's a large panel of scientists from across the world discussing this technology. Kind of trying to figure out what we want to do with this technology, what different things we can do with it, what things do we want to stay away from. Maybe something like designer babies, it's a little scary thing to think of. We probably don't want to be messing in that. This is my opinion, of course, I'm just sharing that. We don't want to be diving into those different things. It's some very ethical issues. But something like plant sciences, this is a great opportunity for us to really take these yields, really take these new great traits and launch them into the next generation. So with that said, I'd like to end my presentation. Everyone, thank you very much for joining me here. Thank you everyone who listened online, and you guys have a great day now.